So Anton van Leeuwenhoek had he was just very, very interested in pretty much everything. And as a hobby, he would produce these very small microscopes where he just basically grounded glass and applied it to very small wireframes. So in doing so, he was just inquisitive about everything. He wanted to see what all of the plaque between his teeth used to look like, and of course they didn't know what his plaque back then. He was interested in rainwater, pond water, anything that he essentially wanted to know about. So as he constructed or made these very small single lens microscopes, he began to view specimens under them, and he was the first to observe, or at least recorded to have observed, what he referred to as animalcules. And animalcules were what we know today as bacteria and protozoans. So you could do this now with microscopes that we have. But if you just took a toothpick and scraped away some of the plaque that's residing around the gum line of your mouth, and if you place that on in a drop of water and then tried to view it with a magnifying glass, or primarily with a microscope, you would be able to see some of the bacteria that Antoine van Leeuwenhoek was able to see. If you took a drop of pond water and you used a simple magnifying glass at home, you'd be able to see some of the microscopic protists that were swimming around in the water, as Leeuwenhoek was able to see. In South Carolina right now, if you take a sample, you might also see very small larvae that are residing in there. So this really brought to life the idea that there was a whole nother world out there. Now, even though Antoine van Leeuwenhoek was able to discover this, he took the initiative to have his animalcules published in the Royal Society. He published them in the form of letters to the Royal Society of London, which he wrote, kind of highlighting what it was he had found and providing descriptions of what it was he had observed by drawing those. So in doing so, what he was kind of generating was kind of this evidence that existed that he didn't really know it at the time, but there was all these small little organisms that which could have strong ties to causing disease. So he's kind of laying the foundations for that at the moment, as we discuss him. So unfortunately, during his time period, there's a kind of long-term perspective that held steadfast, which was the idea of spontaneous generation, where you essentially have life being generated from non-life. But Leeuwenhoek's work started paving the road to the new theory of biogenesis, where life can only come from life. And so another kind of advocate of this theory of biogenesis, or whose work advocated for the theory, was the man by the name of Louis Pasteur, a French chemist, who made significant contributions to the field of microbiology. He was just incredibly inquisitive as well, and he investigated different research projects that focused on the examination of fermentation, which is a process where you have sugars being fermented and they generate waste products of alcohol. And in doing so, this helped to jumpstart the wine industry when Louis Pasteur started manipulating the process of fermentation and kind of exploiting its products to produce beverages. He also gave rise to the process which is named after him, the pasteurization process, which we use to heat our foods. We use it to warm milk. We use it to kill organisms that reside in substances that we intend to consume. And so he was a significant contributor, just contributing those two things, research on fermentation, processes, and then research on the research and the development of pasteurization. He also uncovered or found out about the discovery of life forms in which they can live without oxygen. So these particular type of organisms are referred to as anaerobes. And all of this makes incredible sense because anaerobic organisms are organisms that undergo the process of fermentation because they don't require oxygen to thrive and metabolize or jumpstart their metabolism. So as he's investigating the alcohols being produced through the process of fermentation, he's also coming to an understanding that there are different organisms out there that are not like us. They don't have to breathe air in order to generate energy. So this is obviously very early on microbiology, so there is a limitation to the amount of science that they knew regarding cell molecular structures as well as the processes involved in generating energy inside of a cell. But being able to discover that there are organisms out there that do not thrive or are not able to utilize oxygen in order to live was a pretty significant breakthrough. In addition to those particular achievements, Louis Pasteur was also very savvy in the production of vaccinations. He produces vaccines for both rabies and anthrax. He also, some other attributes that are highlighted by Louis Pasteur is the fact that he was able to study reasons why certain industries were being disrupted from other microorganisms. So the silk industry was hard hit through silkworm diseases, and he was able to figure out how to prevent silkworm disease from happening, so that way silk continue to be produced. He also helped to assist in minimizing the spread of diseases in certain hospitals. He was able to help to kind of, control and isolate individuals that were carriers or, infected with certain agents, to minimize how pathogens were being spread in those particular environments. Now, because of all his great achievements, in 1888, France decided to implement the Pasteur Institute, which is a pretty significant institute, and there's multiple locations all around the world, but the primary one, or the first one, was developed in 1888 and named in Louis Pasteur's honor. Our next pioneer in the field is Robert Koch, and this was a German physician who was also quite the inquisitive person. He essentially gave rise to the germ theory, which states again that disease comes from living organisms, and it just isn't produced out of thin air or through the process of abiogenesis. Life has to come from life. He also kind of studied Bacillus anthracis, which many of you, if you haven't heard before, can be a very deadly organism because it produces spores that can be inhaled by a human, and once they're inhaled, they can be very lethal. So Bacillus anthracis is a pretty dangerous organism. You can also acquire it from not just human to human interactions through exposure to it, but you can acquire it through its natural in the environment. It's on farms, it's on cows. It's a very common bacteria, but can be proven deadly, and if exploited for wrong purposes, it can be used as a biological weapon. And so Robert Koch, some other achievements he came up with were methods for fixing and staining bacteria. When we fix bacteria, we adhere them to a specific slide so we can view them under the microscope. You can fix them in alcohol. You can fix them through a heat fix smear. And when we stain them, we use reagents such as methylene blue, crystal violet, safranin, malachite green. There's all kinds of different stains you can use on bacteria to help enhance their view. Because if you don't stain them, they're just going to look clear under the microscope because they're so incredibly thin and light just will pass through them. So he developed those processes in the sciences. He also initiated methods that were used to cultivate bacteria. So these were methods that are used to help grow bacteria. He used some of the earliest, first known forms of petri dishes by simply using a very flat glass dish. And then he developed the idea of media, which is kind of a substance that's used to provide nutrients to the bacteria that you're interested in growing on it. And he used agar, which is a specific type of large polysaccharide that's taken from seaweed to help produce that media. And we use all of these techniques, the staining, the fixing technique, as well as the cultivation technique to this day. So we have him to thank for those processes. Now, in addition, Robert Koch was very interested in how, disease was caused by living organisms. So he took pretty much the lead in destroying the theory of abiogenesis by doing an experiment with rats. And I'll come back to this slide, because this rat experiment gives rise to what we call Koch's postulates. Now, in this experiment, what you have to do is you have to inject, well, first of all, you have to isolate potentially pathogenic organism. So an organism that has caused disease inside a host, you have to isolate that in the laboratory from a diseased animal. Okay, so here you have a diseased rat. You're going to isolate specific bacteria from that rat that you think is causing the illness. And then you want to cultivate this specific microorganism. Once it's cultivated and joined in a pure culture, you want to take it and then you want to inject it into a healthy rat. And then watch to see if that rat acquires the same disease that rat 1 did, or whoever the patient was where that particular isolate came from. So once you monitor the newly injected host, you also then want to cultivate the pathogen that you injected here and look at it underneath the microscope. Once you observe it under the microscope, you compare this finding here to this finding here. And if they're similar, then you'll be able to see that, yes, this organism caused sickness in this animal. So Robert Koch played around with this quite often, and what he came to conclude was that the microorganisms were inducing infection in healthy patients or healthy animal host. So this gave rise to a number of postulates that Robert Koch generated. And so the first postulate says a particular microbe must be found in all cases of the disease and must not be present in any healthy animals or humans. So if you want to figure out if a pathogen is linked to a disease, you have to check all of the hosts, and all of those hosts must contain the pathogen, and any healthy host cannot demonstrate any of the pathogen once cultured.